Hello everyone, my name is Wang Fu. I come from the Department of Histology and Embryology, School of Basic Medical Science, Shandong University. Today, the topic I will introduce is differentiation of mesenchymal stem cell. Mesenchymal stem cells, abbreviation MSCS, are mesoderm-derived multipotent stem cells that are capable of self-renewing and differentiating into functional cell types. Compared with other stem cells, the ease of isolation, the high expansion rates, the ability of multi-lineage differentiation, the high migratory capacity, the ability to avoid allergenic responses after transplantation, and no ethical issues in autologous stem cell transplantation make the MSCS attractive candidates in regenerative medicine. Over the past few years, MSCS have been isolated from various tissues and organs, including adipose tissue, bone marrow, placental tissue, amniotic fluid, umbilical cord blood, dental pulp, the liver, and the lung, etc. In addition to the extensive sources, the multilineage differentiation of MSCS has been extensively studied in vitro and in vivo since their first discovery, and lots of studies have demonstrated that the MSCS not only have the potential to differentiate into several mesoderm-type lineages, including osteogenic, chondrogenic, adipogenic, and myogenic lineages, but also can trans-differentiate into cells of ectodermal and endodermal lineages, and express markers of neuronal cells, hepatocytes, or pancreatic cells, and so on. During the differentiation of MSCS toward a specific cell type, there are a multitude of stimuli and inhibitors that play important roles in the initial commitment and later stages of differentiation. A large number of studies have suggested that the differentiation of MSCS into specific mature cell types is controlled by various cytokines, growth factors, extracellular matrix molecules, transcription factors, microRNAs, long non-coding RNAs, and epigenetic modification. Moreover, a series of cellular and molecular signaling pathways and microenvironmental changes have also been studied in the differentiation of MSCS. Here, I would like to give an example of the role of transcription factor RANKS2, RANT-related transcription factor 2, in the MSC differentiation. Increased data have shown that RANKS2 is an essential regulator for bone formation and the osteogenic differentiation of MSCS. It directs the MSCS to differentiate into pre-osteoblasts and inhibits the adipogenic and chondrogenic differentiation. Meanwhile, the expression of RANKS2 is regulated by many signaling pathways, including Wnt, BMP, TGF-beta, and Notch signaling pathways. This figure briefly shows that MSCS can differentiate into many kinds of cell types, such as osteocytes, chondrocytes, adipocytes, and cardiomyocytes, and so on. Meanwhile, various transcription factors, such as RANKS2 and GALTAS, have been involved in the differentiation of MSCS. Lots of studies have shown that the deletion or induced mutations of RUNX2 or the corresponding signaling path molecules can disrupt osteogenic differentiation of MSCs, while the delivery of transcription factor or signaling molecules into MSCs can increase and maintain the potential of MSCs to differentiate into intended cell lineage. This figure illustrates the effects of Wnt signaling on the various MSC differentiation pathways, and many studies further demonstrate that the activated Wnt protein functions through the canonical or non-canonical Wnt signaling pathways in the MSC differentiation. To date, we have achieved great advance in the differentiation of MSCs, and major breakthroughs have been made in cardiovascular disease, neurological disease, autoimmune disease, and bone injury repair, etc. However, there are still many challenges to fully understand the mechanisms underlying the differentiation of MSCs in various lineages, including the identification of the signaling molecules, transcription factors, as well as the microRNAs, non-coding RNAs, and the crosstalk between signaling pathways that functions in the differentiation of MSCs. With the deepening of research and the increasing of clinical trials, MSCs show great basic research value and broad clinical application prospect, and represent a promising source for regenerative medicine. That's all. Thank you. See you next time.